Hey, good afternoon, everyone, and thank you so much for joining us today. My name is Matthew Anderson. I'm the CEO of Vanguard Scientific, and today I get an opportunity to interview and speak with one of the brightest minds that I've met in genetics and biosciences surrounding active botanics and ingredients, the Chief Scientific Officer and co-founder of Mydecine, Robert Roscow. Mr. and Dr. Roscow, how are you today, sir?
Great. It's great to talk with you, Matthew, today. So really excited to be here.
Yeah, we had about 15 minutes to catch up earlier, and I got a fire hose, to say the least, on the perspective you have on psychedelic, psycho-assisted pharmaceuticals and therapeutics, what you're doing in drug design, what you're doing in breeding, and just overall research is inspiring. I would try to give an elevator pitch of what Mydecine's doing. You can go to your website. You're a publicly traded company, but we have the opportunity to hear it from you. I'd love a quick elevator pitch, and then maybe let's answer some questions back and forth.
Sure.
Rob?
Absolutely. So thanks for giving me the opportunity to talk about Mydecine today. And I'll really start by, explaining why we founded Mydecine. And really it's to take advantage of the fact that there's really a sort of trifecta brewing around the interest in potent medicines from fungal biosources. And so if you think about that, it's really sort of started by the fact that there's a huge increasing amount of clinical data coming out about the possible effectiveness of the classical psychedelics, specifically psilocybin, as applied to really intractable, currently underserved mental health conditions. And so that was one facet of it. Next facet is that the clinical data is actually supporting that. So you have the public interest because of the clinical data. The clinical data is really also getting stronger at the same time. So scientifically, this is much better supported over time. And then third, that because of these first two factors, the investment community in the biotech space also got very much interested. So we had this sort of, perfect storm of public interest, scientific know-how and backing to feasibly do these things, and then also the monetary resources to do this becoming available. And so we really thought, this is the perfect time to drive forward on really developing drugs in this category. And if we, think about kind of our prime indications of interest at Mydecine. So, fast forward from that inception, we've really itemized both severe PTSD in a veterans population and then addiction as prime targets of interest. And so if we think about, how do we go about designing a program to design drugs in that category, really what we're doing is thinking about, okay, well, those conditions, what pathways, what neurological systems are really at play, and then can we actually find active components that are acting as effectively intermediaries in that pathway to then cause signaling. And so this is what we've done in the fungal space, where we've looked specifically at psilocybin, and then in a drug development context, how would we actually take that potent template from nature? Again, this is what was kind of building that perfect storm. But how do we take that and improve it for better safety or efficacy in a clinical context? They're not perfect solutions in nature and can definitely be tailored to either better match clinical practice or to reduce already known safety side effects, et cetera. So that's really the inception and goal behind Mydecine in general, is take these potent molecules out of nature and then bring them along with best practice, safety, and efficacy into the hands of clinicians.
I love it. First off, you can tell that you're an entrepreneur because you can deliver the elevator version, and then you can also get into the details. Anybody that's watching, you've got a recording of this video, I encourage you to watch that again. It's really refreshing to talk to a chief scientific officer. Being a business guy myself, I don't have those capabilities, but the way you just explained the work that you're doing really gives anybody watching an understanding of the level of detail and the discipline that it was required to begin to do some of this work. You mentioned drug design.
Mm-hmm.
It's not typically something that's talked about in emerging investment opportunities or in new companies that are entering markets, especially in new categories. What does it take in order to develop a drug? And specifically in this case, a psychedelic drug made from psilocybin, sure, but I'd just love to hear it from you.
Absolutely. So first, to develop a drug, really you need a target. You need a disease with a known pathway, known neurological or cellular pathway to target, because this is really what you're doing with a drug. You're finding a compound that's very similar to a compound in the body that then will have a medical effect. And so if we think again about our core area of interest around the mental health conditions, specifically PTSD and addiction initially, what we're really looking at there is historically there's been a strong interest in the serotonin pathway and serotonin-based drugs. You see this in the sort of first generations and second generation of antidepressants. But what we can see with psilocybin is actually a almost exact match for the native serotonin. And so we know that we have a molecule from these fungal sources that will activate this pathway. And the question then becomes, how can that be, if we are activating the pathway, how can that be leveraged for medical benefit? It's not a one-to-one transition. There's a number of things that need to be looked at as to how are you providing that? What is the correct dosage? Are there adverse events or side effects that you may want to ameliorate or deal with? And these are all things that can be addressed with either making novel iterations off of the base template, including them in complex formulations or at different purity levels. All of these different questions can be addressed with sort of those three techniques. And so as you think about that, okay, well, we have a potent serotonin analog that's going to activate the serotonin system. And we know that it may provide benefit to therapy. So how do you bridge that gap? And really what we're doing to bridge that gap is controlling dosability, controlling side effects, and then thinking through in a therapeutic context, how do we best match therapy to the actual drug effect? And this is kind of the storyboard timeline of what we're doing to develop these drugs. And as each of these stages can be really enhanced by knowledge of the field, knowledge of therapy, knowledge of pharmacology, knowledge of the underlying chemistry of the tryptamines, this category of drugs. And at each point, we can kind of make these iterative improvements.
It's incredible. And again, I love the way you articulate it. You said beginning to control side effects, right? Begin to control half-life or dose. Those are not light lifts, and answering some of those are some of the largest questions I know you tackled in cannabinoids at your work with 𝗙ance and every prior. But to hear the conversation now really moving into a pharmacological setting around specific molecules is incredibly interesting. I'd love to go just a little bit deeper there. We were talking earlier, you had shared about some of the work you were doing with Dr. Brian Reed. And some of that predicate work that's leading you to take apart, let's call it lion's mane or psilocybin, and beginning to look at the specific molecules that could eventually mature into drugs. I'd love you just to talk a little bit about that, because it was so cool to hear.
Absolutely. So yeah, my prior work was very much based on patentable formulations and specific outcomes of purified cannabinoids and terpenes as mixtures. And so this is a analogous problem to what we see with developing medicines out of fungal sources, because any of these biological sources are not just a single compound. They're groups of similar related compounds, as in cannabis, where you get a number of cannabinoids. In terpenes, it's the same in lion's mane. You get a number of ericinones and hyrisines, these larger families of molecules. And so it's at a basic level, it's kind of the same question. So really what my work back at 𝗍𝘀 and then 𝗙ance with Dr. Brian Reed was really highlighting is a great way of how we would actually tease apart this complexity and then actually build back a product, a medicine, et cetera, with the properties that we want from that complexity. And really what we're looking at is you take, so in this case, whole psilocybin mushrooms, and we can actually strip them down to not just the purified psilocybin, but all of the small minor tryptamines. There's a number of other tryptamines similar to psilocybin that are produced. There's a number of other molecules that possibly may interact with those tryptamines. And so what we can do is purify them all to their single entities, start looking at these on benchtop experiments that give us the output, the cellular output of the strength of these. And then we can also start making combinations. So we have kind of a benchtop output. And then we can then take that and make hypotheses about how that may work in either an animal model system or in a human down the line. And so these are really the building blocks of how do you take something that obviously has medical potential and has been historically used in the case of lion's mane specifically, for thousands of years in East Asia as traditional medicine. And so there's strong likelihood that these are medically very potent, but that complexity is a really big wall compared to how traditional medicines have been developed
Absolutely.
to this point. So it really needs that screening step.
One of the biggest walls, I'd say. I mean, and that's why it was to listen to you unpack that for me and how you walked through that issue, being able to actually track, monitor, and kind of indicate that's otherwise not available. And it's exciting. It only makes sense that it's on the forefront of new pharma and really what's being done there. The real interesting thing about any new business, again, kind of as a chronic entrepreneur, is you have the idea and the vision, and then it takes the requirement to deliver. You talked around the perfect storm timing around capitalization, investor interest, and market. But that doesn't really give you enough credit around the operation of Mydecine. It's incredible to hear you talking about doing these studies and doing these benchtop research. But when you go a layer further, you learn that you have operations here as well as internationally located in Canada. You're doing pharmaceutical grade work. You're dealing in the nutraceutical or supplement space here, which is enabling you to work in this regulated environment. I'd love just to talk a little bit about that, and then maybe some of that predicate experience you got from cannabis and how it helped inform this strategy, because it's really smart.
Absolutely. Thank you. I'll kind of go through the footprint of the company and then explain how we're utilizing that footprint. And so really what we've done is, partly the backstory of this is that we started Mydecine during COVID. So that's always one asterisk to say with this is, that's partly why we're so international. But we've really, itemized what are the scientific resources that we need to run an R&D project, exactly like we've just been talking about. And so we have need for intricate chemistry, intricate pharmacology analysis, and then also for really licensure to control some of those activities. And so one of the first relationships that we stood up with Mydecine as we were founding the company is a strong partnership with the University of Alberta through Applied Pharmaceuticals Innovations. And so what this does is it actually gives us access to basically all of the resources at the University of Alberta. So we have a mature, world, renowned pharmacy school at our disposal to collaborate with. They also then hold licensure to allow us to do a lot of the physical work with these scheduled substances, and that then allows us to put those into clinical trials. If I step back though and think about, the founding principles of the company as far as, how do we actually take medically potent molecules out of fungi and then develop them into over-the-counter or pharmaceutical prescription medicines, there's also a very strong interest, and this is overlapping with our psilocybin interest in some of the functional mushrooms. And so we already mentioned lion's mane. We also have a strong interest in 𝗙𝗄𝗧𝗋𝗃, for instance. And this is really us looking at where there's potent pharmacology in traditional use of these compounds, and then where is there also the best clinical data to overlap. And so we're enabling a lot of this work out of the our own lab in South Denver in Colorado, where I'm based out of. And when I say this is overlapping, is that we're also very interested in production systems. And so we can actually enable a lot of the production systems. Fungi can be very much leveraged in a laboratory cultivation sort of context. And so we can leverage those in the non-psychedelic, non-scheduled species, et cetera, and do a lot of enabling work there that then also has, benefit in, if you look at our interest in these other ones, they are either cognitive boosters, energy boosters, and, VO2 max modulators, or sleep aids. And so there's, strong support of these also across. And so with the resources that we have to look at these again in a drug development, context, it's really been I think, situation where we set up a unique footprint of resources for addressing these questions. I look around at our competition, the other people in the space. It's a really exciting industry right now, but I don't see anybody really looking at these from the same lens or the same sort of angle as we are, or with the same resources.
Yeah, there's working hard and there's working smart. There's no doubt there's opportunity on the horizon, but how? And you just said it. So, again, it's awesome to allow the opportunity for the viewers to understand the scaffolding and the structure of what you're doing, why you're doing it, and actually how you're delivering it. The resourcing is not light, and in drug development, entities. It is the research pipeline and the why. And again, for you to be able to break apart the operational costs, the performance sequencing, and then the reasoning with overlaid research is another reason why I'm just continuing to get more and more excited about Mydecine. Rob, we've got about 2 minutes left at the most. Is there anything else you'd like to cover with the group today?
I think one thing I'd like to cover, yes, is really, I think, if you compare Mydecine to other drug development companies, one of the things that I think is really a strong suit for us and is uniquely different is the fact that we are not developing drugs out of thin air. We're not trying to just design novel molecules with the hope that they will hit our receptor systems of interest, our biological systems of interest. We're really looking for where is there the best evidence across human history, because the traditional use of a lot of these compounds is quite deep. And where do we have potent molecules that then we can do exactly the work that we were describing on? And I think this is a really risk-averse strategy in drug development, where the common conception is that there's a lot of failure for each sort of molecule tried. But if you have this backlog of already demonstrated success, that risk is very much removed. And so I think that's a refreshing strategy for me as a scientist to follow.
Rob, it was an absolute pleasure talking with you today. Again, the chance to speak with someone that's carrying the scientific mind as well as the business strategy is inspiring. At the forefront of a new enterprise and industry, congratulations on Mydecine. It sounds like you've got the work cut out for you, but I suspect you're going to find the support you're looking for. Thanks again for sharing time today.
Oh, thank you again. It's been fun talking.
Have a great day.